Start with the name of Allah, who is most merciful and beneficial. Today we will discuss the bacterial genus Proteus. This is the shape of bacteria. Let's talk about history. The Proteus bacteria was first discovered by Gustav Haster in 1885. The term Proteus was first used by Haster in 1885 when he was determined three types of organisms in the microscope when he was studying the putrefied meat. The term Proteus means changeability of form. It means this bacteria have specific characteristic that it changes form in liquid and solid media. So that's why it is also shown the name the old man of sea. This is the classification. This group of bacteria have family Enterobacteriaceae and genus Proteus. These all the species of Proteus are not so much important, but two species are more important: Proteus mirabilis and Proteus vulgaris. Let's see general characteristics. It is a gram-negative facultative in nature. Facultative means this bacteria grows in the presence or absence of oxygen. Actively motile, possessing peritrichous flagella, reducing nitrates to nitrates. It means it is a saprophytic bacteria. Saprophytic bacteria are those which live on the dead materials or dead organisms and gets nutrients from that dead or declined nutrients or plants. This group develops as slowly as the temperature 0 degrees Celsius and as high as the temperature rises up to 43 degrees Celsius to 45 degrees Celsius. This is the structure of bacteria. Morphology. It is a bacilli, highly motile, very leomorphic, means highly motile due to its flagella, and leomorphic mean it have more than one form. Cells are short, colli like rods with rounded ends. In liquids, its form is a swimming, just like something swimming on the water or liquid have the size 1.5 to 2 microns and which contains 6 to 10 flagella. On solid media or solid surface, it forms much larger cells than the swimming form and have 60 to 80 microns and exhibits thousands of flagella. Mean this form is larger than the other form, swimming form, have multinucleated cells. This is the nutrient agar structure which form the swimming cells. Let's talk about its culture characteristics. On the nutrient agar, it forms colonies with swarming growth in successive waves and fishy odor. Swarming growth, as we tell later, that swarming growth is the growth which contains circular movement of the bacteria due to its ability of the changeability of form. On the blood agar, it forms no colonies. Instead, bacteria swarm on the agar surface. In the blood agar, it forms swarming growth, but no colonies. In the McConkey's agar, it forms non-swarm, smooth, pale or colorless colonies. The blood agar and McConkey's agar are the most important for these types of bacterial growth. Let's talk about most important biochemical reactions. First, urease test. Urease test is a test in which we determine the bacteria which release the enzyme urease or not. So this bacteria is urease positive, which means this bacteria release enzyme urease. Urease is the enzyme which convert urea into ammonia and carbon dioxide. Second test is TSI, mean triple sugar iron test. In this test, we use three types of sugars: number one glucose, number two lactose, and number three sucrose. And we see its pH on the upper and lower side of the test tube. So this test is positive, means the Proteus bacteria utilize these sugars and the pH is alkaline acidic. Next test is indole test. So indole test is the test in which we see the bacteria is converted the phytophthalein into indole or not. So this test is positive, but one species Proteus mirabilis is not, is negative for this test. Next test is phenylalanine deaminase test. In this test, we determine the bacteria is producing deaminase enzyme or not. Deaminase enzyme convert or break the amino group in the phenylalanine compound. So this test is also positive because this Proteus bacteria produce a deaminase enzyme. So these are all the biochemical reactions we see. The next is pathogenicity of the Proteus bacteria. First, this bacteria enters into the cell due to its body and moves into the cell with the help of flagella. Due to its production of urease enzyme, which converts the urea into ammonia and carbon dioxide, the pH of the urinary tract is high up to alkaline and metabolism also disturbed. So due to the urease production, it causes many inflammation in the urinary tract. Among all the species of bacteria, Proteus mirabilis is highly pathogenic, which causes many diseases like urinary tract infections, community pneumonia, surgical wound infection, and postoperative endothelmitis, bacterial endocarditis, and some show some symptoms like cystitis, urethritis, prostatitis, pyelonephritis, etc. These are the symptoms which are seen in the urinary tract infection because this bacteria mostly infectious to the urinary tract infections or UTI. We discuss some diagnosis. Diagnosis purpose, we use different types of samples. Number one is blood sample, sputum sample, urine sample, swab sample. These are all the samples which are used for culturing and biochemical tests. These are the treatment for the bacteria: fluoroquinolone, cephalosporin, gentamicin, and ceftriaxone. Cephalosporin is some resistance to these bacteria, so this cephalosporin is not commonly used. The commonly used drug is the fluoroquinolone, which are effective against the P. mirabilis and P. vulgaris, which are the high pathogenic to urinary tract infections. So these are the some aspects of genus Proteus. Thank you very much. Allah Hafiz.